Okay, can somebody confirm you can hear me?
Yes.
Yes.
Thank you. All right, are there any questions before I begin? Let me state that I have done some grading. Let me stop the share. Then I'll call up the grade book and then I'll have an easier time talking about it. So I can shut that down. So I can shut that down too. So I've got the Pelagie quiz graded, the extra credit picture graded, the extra credit study sheet graded, the extra credit study 2 graded. Worksheet for lecture 1 and 2 graded. And it looks like there's somebody I need to grade for lecture 1. I'll take a look at that. The quiz 1C is graded. I haven't really gone in and looked at it. I'll have to look at that a little closer. And then the first grading of lab 0, lab 1, and lab 2 is done. I have not graded any of them a second time. I have not graded most people's practice quiz, either 00A or 00B. I have started on that. I think I started on, I don't even remember if it was 00A now or 00B. But most people are not graded on that. I'm working on that. Any questions about the grading? So take a look at your grade. For the labs, remember for lab, is it lab 2? Yeah, lab 2. If you didn't get 100%, you can submit lab 2 by this Saturday, and it will be for full credit. All right, let me get out of the grade book and then I can share my screen. All right, today is January 25th, and we're going to be discussing lab 4 on stains today. Any question about that? If not, let's go to the lab. Lab 4, staining techniques. We're going to talk about the simple stain, the differential stain, and special stains today. You do need to read some material in the textbook. You can read about the size, the shape, and the arrangement of bacterial cells. You can read about the bacterial cell wall. You can read about the simple stain. You can read about differential stains. Of the differential stains, there's the gram stain and the acid-fast stain. There are other differential stains, but we're not going to discuss any others. And then you can read about the special stains. We're going to talk about 3 different special stains. Special stains are stains used to stain special cell structures. We'll talk about the negative stain for staining capsules. It's also called the capsule stain. We're going to talk about the stain for staining endospores and the stain for staining flagella. Now, all of these are just a couple of pages to read. This one's only 1 page. That one's only 1 page too. That one page. In today's lab, there are video clips and a virtual lab. Let's cover the learning objectives for this lab. 1, be able to prepare a bacterial smear for staining. 2, understand the principles and procedures used when performing a simple stain, a differential stain, and a special stain. 3, be able to interpret the results for the simple stain, a differential stain, and for the special stains that we're going to talk about. And remember, there are 3 special stains we are covering. And then 4, be able to define the following terms: simple stain, differential stain, special stain, coccus, bacillus, spirillum, peptidoglycan, gram-positive, gram-negative, acid-fast, meaning the acid-fast stain, primary stain, mordant, decolorizer, counterstain, capsule, endospore, and flagella. Any questions about what we're going to cover in the learning objectives? All right, bacteria are essentially transparent. Therefore, in order to see them well under the microscope, they need to be stained. How you stain cells is first, you make a bacterial smear. How you make a bacterial smear is you put a drop of water on the slide. Then you add your cells in a loop, pretend this is a loop, and you only add a little bit of cells, and you mix it to the drop of water. You then smear the drop of water all over the surface. Actually, before you do that, in the drop of water, you mix your cells in the drop of water. And then you smear the mixture all over the slide. That is to spread out the cells and the water so that the slide will dry quicker. You then allow the slide to air dry, and then you heat fix the bacteria onto the glass by just gently heating the slide. If you do not air dry and heat fix the bacteria onto the slide, the bacteria will be washed off in the staining process, which happens after you made the bacterial smear. So let me reiterate, a bacterial smear is something you do before you do the staining. It is a separate event to make the cells on the slide. It is not part of the staining procedure. It is independent. All right. Simple stains is a technique that uses one dye to stain bacterial cells. You can determine some things from this, such as the shape and the cellular arrangement, how the cells are growing together, from the simple stain. But you cannot tell differences from the cells except for if they have a different shape or cellular arrangement. A differential staining technique usually uses 2 or more dyes to provide additional information about the cell, and it can differentiate between 2 different cells. Lastly, special stains are used to dye particular bacterial structures, and we're going to talk about 3: staining for capsules, for endospores, and for flagella. And these are called the capsule stain or the negative stain, and the endospore stain and the flagella stain. When we look at cells under the microscope, we can notice that they have a certain shape, that's a morphology, and they have a certain cellular arrangement or cellular growth arrangement, and that's how the cells stick together. So most cells are cocci or bacilli. There are some that are also spirilla. So these are the three major shapes that bacteria have: cocci, bacilli, and spirilla. Cocci are spheres, bacilli are rods, and they're sometimes just called rods. And then spirilla are spirals, like a gentle S shape. Now, not all, most bacteria, if they're not one of these three, they're a combination of one of these three. Like a coccobacilli is a combination of a cocci and a bacilli. There are a few cells which are pleomorphic, and these species, when you stain one species, you will see the shapes of the cells differ, like these here. And this is not very pleomorphic. You can get much more pleomorphic than this. But that one sort of looks like a footprint. This one actually looks like a rod. That one looks like a gentle spiral. And these look like curved rods. Okay. And you can get them in much more diverse shapes than this. There are a few other shapes too, but we're not going to cover that at least now. But the three main shapes are cocci, bacilli, and spirilla. The growth arrangements are the way the cells stick together. The first growth arrangement you can have is not even shown here, or maybe it's shown there and there. And that is you can have a singlet where the cells are one cell and they're not stuck together. With cocci, you can also have two cells stuck together, and that we call diplococci. Diplo means two, cocci means cocci, two, a sphere-shaped cell. You can have tetrads, four cocci stuck together. You can have many cocci stuck together, and this we call staphylococci. You can also have the cocci in a row, and this we call streptococci. You can have the bacilli in some of the cellular growth arrangements, but not all of them. You can have streptobacilli. You can have diplobacilli. You cannot have cocci, you cannot have tetrads of bacilli. I don't think you can have staphylococci either, but you could have a clump, and a clump is probably not the way the bacilli are growing. It's just they're stuck that way. And I'll talk about that when we get to that in the lab. We won't talk about that now. Any question about any of that? Sorry, I shouldn't have mentioned that. All right, let's talk about the differential stains. The most important and the most commonly used differential stain in microbiology is the Gram stain. This is a stain invented in 1884 by Hans Christian Gram. You do not need to know the year he invented it, but you should know that it was Dr. Gram, Hans Christian Gram, who did invent it. And whenever we spell the Gram stain, that should be a capital G, because the stain is named after Dr. Gram, and we always capitalize a person's name. Dr. Gram invented this stain to differentiate between bacteria cells, which stained dark purple, and then the patient's own cells, which stained pinkish or reddish. We'll say pinkish. Microbiologists started using Dr. Gram stain, and they discovered that this stain was a very good stain. and it could differentiate between two very large groups of bacteria. The gram-positive cells, which stain dark purple, and the gram-negative cells, which stain pinkish. And the gram stain distinguishes bacteria according to their ability to retain the crystal violet, the first dye used in the gram stain. The first dye used is called the primary dye. Bacteria that retain the crystal violet, or the primary dye, are called gram-positive cells, and they appear purple, while bacteria that do not retain the crystal violet, meaning they lose the crystal violet, are referred to as gram-negative, and they appear pinkish. So here we have a gram stain of many different species of cell. You can have, let me blow this up. There's the gram-negative bacillus, a gram-positive bacillus, a gram-negative spirillum or spiral right there and there. I think I already said a gram-positive bacillus, but there's one there. And there are some gram-positive cocci. I just noticed there's some cocci there, but I can't make out the color. Let me see if I can blow this up even further. I think these are gram-positive cocci, but they're not probably in good focus, so they're not showing up too well, or maybe they're not staining too darkly. But all the cocci here and here look like they're gram-positive cocci. Ooh. All right, the gram stain differentiates between the bacteria based upon their cell wall. The gram-positive bacteria is structurally different from the gram-negative. The gram-positive bacteria have a very thick peptidoglycan layer, while the gram-negative cells have a peptidoglycan layer, but it's much thinner. When we look at the bacteria, you can see the gram-positive cells here, and there's the plasma membrane, there's the cell wall, and the cell wall of the gram-positive cell is mostly this peptidoglycan layer, and it's very thick, around 100 layers of peptidoglycan, one on top of the other. And if I blow this up enough, you can actually start to see some of the layers here. So that would be one there, and a darker one there, and one there, and one there, and one there, and one there, one there. And then there's one here, and this is probably several layers. The point is, there's many layers of peptidoglycan in the gram-positive cell. Now, when we come over here, we can see the gram-negative cell, and it has a cell membrane as well. And outside the cell membrane is the cell wall. There is a peptidoglycan layer in the cell wall, but it's much thinner than the gram-positive's peptidoglycan layer. And then outside the peptidoglycan, we have a lipid bilayer that's called the outer membrane of the cell wall. And we'll talk more about that in another lab, a later lab. We might talk about it next week, but I don't remember if it's next lab or we'll soon talk about it, but we're not going to talk further about the lipid bilayer called the cell membrane of the outer of the cell wall. We will talk a little bit more about the peptidoglycan layer, both today as well as I think our next lab. Oh, I forgot to mention that the textbook, when they show you the gram-positive cell, they only show 3 layers of peptidoglycan. And in the gram-negative cell, it's even worse. They only show 1 layer of peptidoglycan. And the peptidoglycan is in the gram-negative cells, at least 3 layers, up to about 20 layers of peptidoglycan. So it's much more than 1. And then the peptidoglycan layer in the textbook is only shown as 3. And as I stated, it's mostly around 100 or more layers of peptidoglycan. Many layers of peptidoglycan. All right, so in the gram stain, cells are stained first with the crystal violet, and it will stain the cells a bluish purple color. We then stain the cells, not stain the cells, we then apply Graham's iodine. And Graham's iodine is a mordant here. A mordant is a chemical which combines with the crystal violet to form the crystal violet iodine complex, which is larger than the crystal violet or the iodine alone. And that's just to make a larger molecule so it's won't wash out as easily, particularly in the gram-positive cells. And then next in the gram stain, we treat the cells with alcohol acetone, which is used as a decolorizer, and that will remove the crystal violet iodine complex from the gram-negative cells, but not from the gram-positive cells. And the reason is simply the thickness of the peptidoglycan layer. In the gram-negative cells, the thickness is much thinner than in the gram-positive cells, so that the crystal violet iodine complex comes out in the gram-negative cells, but does not in the gram-positive cells, at least if you apply the gram stain correctly. The gram-negative cells become clear, and then we stain the cells with safranin, and that will make the gram-negative cells pinkish in color. The timing of decolorization is critical, and I'll talk a little bit more about that later in this lab. All right, any question about any of that? Some bacteria do not stain well with the gram stain, and that is mainly members of the family Mycobacterium. The Mycobacterium have a lot of macolic acid in their cell wall. Macolic acid is a lipid, and this lipid resists stains and other material which dissolves in water. The gram stain is dissolved in water, and so when we stain the cells, members of the family Mycobacteria tend to stain lightly. And there might be a few that don't even stain, but to all my knowledge, and I've really only worked with, I think, 2 Mycobacteria, maybe only 1. But anyways, it does stain lightly in the gram stain, meaning if you gram stain the Mycobacteria, they do pick up the gram stain, meaning the crystal violet, and then they retain it. So they are gram positive. And they are gram positive by the structure of the cell wall, meaning it's a thick peptidoglycan layer. However, there are other things in the cell wall like mycolic acid, which are not normally seen in other families of bacteria. So mycolic acid is only found really in two families, the Mycobacteria and the Nocardia. All right, any questions about any of that? Oh, let me state, because the mycobacteria do not stain well in the gram stain, they are usually staining lighter in color than another cell in a family other than the mycobacteria. Anyways, the mycobacteria, when they're stained, sometimes won't stain well, or they will be variable like this. This is one species, and it was stained by the gram stain. And as you can see, we have two different colored cells, which look the same. And that is a gram-negative bacilli staining pinkish, and then gram-positive bacilli staining purple, dark purple. What do you call these, gram-negative or gram-positive? Well, you can call it mixed. The mycobacterium actually are gram-positive. And the reason why these cells are staining variable, meaning some gram-negative and others gram-positive, is because this is an old culture of cells. If you use the gram stain, you should only use it on cells which are 2 days old or less. And that's because you do need intact cell walls and viable cells in order for the gram stain to stain correctly. The gram stain is not staining well here because there's two different types in one species. And the reason is because we're not using freshly grown cells. These cells were always older than 2 days old. And actually, the only time I've ever seen gram variable in microbiology was when the student called me over and said, "Look at my slide here. Why are some of my cells pink and others purple?" And I asked them, "How old were your cells that you used to make the gram stain?" And in every case, the student said, "Oh, 2 weeks or more." And I'm telling you, you can't use old cells in the gram stain, or the gram stain may give incorrect results. The reason why the cells are dark purple here, and then they have a very distinct border, is because these cells are viable. On the other hand, what do you notice about these cells here? This cell here, this cell here, and particularly this cell here, the pink one, right there, the little one, not the dark one or the big one. What do you notice about those cells? Anybody notice anything about those cells?
The borders look a little less defined.
Yeah, the borders aren't sharply defined. This cell even looks like it's broken. And that cell is staining differentially. The very top, very light, very skinny. Down below, the cell is thicker and darker. It's all pink, but it's definitely different. And the reason is because some of these cells, because the cells are so old, Some of these cells are dying and the cell wall is deteriorating. And this cell wall is broken in two. That's probably why that cell is so funny looking, white, skinnier on top and thicker below. And the cell wall is probably just disintegrating. And these cells, the gram negative, are probably dead or dying cells. And because of that, they're not staining correctly. If you have viable growing cells less than 2 weeks old, you won't have this, at least in my experience. And that's because I told the students, I asked them how old it was, and every case the students told me, oh, these cells are 2 weeks older or older. And I said, no, you can't do the gram stain on old cells. Otherwise, the results could be wrong. And of the students who repeated the experiment, they had to grow up new cells and then repeated the gram stain. They never saw gram variable. They only saw the members of mycobacteria to be gram positive. Any question about any of that? All right, there are some tips for preventing your cells from being gram variable. First, and most importantly, avoid using old cells. Old cells are dying cells, and they will not stain correctly with the gram stain. Prepare a thin smear. The ideal bacterial smear is when the bacteria are a single layer thick on the slide. If there are multiple layers of cells on the slide, it's going to be very difficult getting the dye all the way through the cells to stain the cells on the bottom. And then conversely, so the bottom cells may not pick up the stain. They certainly may be lighter than the cells higher up in the smear. And the other reason is the thick layers of cells will be difficult for the dye to get down through the layers to stain the bottom layer. And it will also be difficult for the decolorizing agent to get through all of the layers of cells and then decolorize the bottom layer. So prepare thin smears. Then heat fix the bacteria onto the slide only after the smear has air dried. If you heat fix the bacteria before the cells air dry, you will cause water to evaporate, and that can come out of the cell, destroying the cell wall. Avoid overheating the cells during heat fixation, because the heat can disrupt the cell wall as well. Obviously, use fresh staining reagents to prevent a gram variable reaction. And then another very major important one is do not over decolorize. If you over decolorize, add too much decolorizing agent for too long a period of time, you will pull out the crystal violet iodine complex from the gram positive cells. You should also avoid under decolorizing. If you do not decolorize sufficiently, not all of the cells will have the crystal violet iodine complex removed, even though they are gram negative cells and the same species member as the other cells, which are staining a given color. Any question about any of that? All right. Let's stop talking about the gram stain and briefly talk about the acid-fast stain. That is the only other differential stain, we're going to talk about some species, mostly in the mycobacteria and the nocardia families, have a waxy substance called mycolic acid in their cell wall. These bacteria are able to resist the decolorization step in the acid-fast stain. And these cells which resist being decolorized, we refer to them as acid-fast bacteria that are positive, or you can call it acid-fast positive bacteria. The cells which give up the primary dye in the decolorizing step of the acid-fast stain, we call those cells acid-fast negative bacteria, or acid-fast bacteria that are negative. Initially, when you're applying the acid-fast stain, the primary stain, the first stain you're applying, all the cells will take up this primary stain. And its name is actually carbonyl fuchsin. It's a cherry red color. However, the cells are then decolorized, and all of the acid-fast negative cells will lose the carbonyl fuchsin. We then apply another dye to colorize the uncolorized cells, and that is usually either methylene blue or brilliant green, but occasionally it's crystal violet. And this stain we call a counter stain because it is the second stain used, and it's a different color from the primary stain. At the end of the staining process, you then take a look at the cells. The acid-fast positive cells will stain red or reddish, and the non-acid-fast cells will lose the carbonyl fuchsin, and then they'll be stained blue or green depending on which counter stain was used. Methylene blue will make the cells turn blue, and is it malachite green? That's not right. Brilliant green. There it is. Brilliant green is the other stain that's used as the counter stain in the acid-fast stain. When we look at the acid-fast bacteria, if the cells are acid-fast positive, they will stain pinkish, and the acid-fast negative bacteria will stain greenish or bluish, depending on what color the counter stain was. All right, let's talk about the special stains. There are three I'm going to talk about. The negative or capsule stain, which stains the capsule. The endospore stain, which stains endospores. And then lastly is the flagella stain, which stains flagella. Let's first talk about the negative stain. It allows the glycocalyx to be seen in a cell. If the glycocalyx is firmly attached to the cell wall, we refer to it as a capsule. The glycocalyx, if it's a capsule, it also has to have many molecules in the glycocalyx besides water. This isn't listed in this slide, but we'll talk about it later, so I'm going to briefly mention the other type of glycocalyx is the slime layer. The slime layer cannot be seen in the light microscope. To see the slime layer, you have to prepare the slide for use by an electron microscope. So the slime layer cannot be seen with the light microscope, but it can be seen with the electron microscope. The capsule can be seen with both microscopes, most importantly, the light microscope. All right, any question about that? I should mention the glycocalyx is something made by the cell, but is excreted outside the cell and is not considered a part of the cell. It's an extracellular material. Productions of capsules can be associated with an organism's virulence or pathogenicity. The capsule can protect the bacteria from being phagocytized. A white blood cell wanders the body or the circulatory system looking for foreign agents, bacteria that shouldn't be in the body. And then the white blood cell can kill the bacteria. And the most common way that white blood cells kill the bacteria is by phagocytosis. They engulf the bacteria and then digest it. When we look at cells that form a capsule, when you're looking at their colonial growth, you note that they look mucoid in appearance if they are mycobacteria and they have a capsule. All right, capsules are not easily observed when using conventional staining methods. For example, the steps of the gram stain will wash away the capsule from the rest of the bacterium. We use a negative stain to visually see the capsule. And usually the negative stain, also called the capsule stain, uses India ink or nigrosin. If the capsule is present, there will be a clear zone surrounding the cell, the bacteria. Let me talk about it here, as this is a negative stain. It's called the negative stain because it's staining the background and not the cell so much. It is actually staining the cells. They're being stained lightly, but they're not being stained darkly. And obviously the background of the slide is stained very darkly. The cells themselves are kind of gray in color, and that's just because the India ink is not good at getting inside the bacteria or even coating the bacteria with the India ink. So the background looks dark. The cell looks grayish in color. And then the capsule forms a clear ring around the bacteria. Anyways, that's the negative stain using India ink. This is a capsule stain, but it is not the capsule stain I just discussed. And I'm not sure which capsule stain that is. Ugh. All right, let's move on to the endospore stain. Endospores are metabolically inactive structures. They're also hardy, and they're produced by some bacteria as a defense strategy when the environment becomes unfavorable. When the environment becomes too unfavorable, the bacteria cell will die. The endospore, on the other hand, will continue to be alive. It's not very metabolically active. It doesn't grow and reproduce. And to my knowledge, it doesn't have any biochemical reactions going on in the cytoplasm proper. But my guess is there probably is some things that happen. And then when the environment changes and now becomes favorable, the endospore will germinate and the bacteria will change from being an endospore to being a normal plant, whatever the plant is, and the cells will become metabolically active. Any question about any of that? All right. I think I talk about it later, so I'll just talk about that later. Most of the bacteria that form endospores belong to two families, and that's family Bacillus and family Clostridium. This table is showing you some pathogenic endospore forming gram-positive bacilli. So the specific species name of the bacteria is on the left here, and then the clinical significance of an infection from this bacteria. So if somebody got a Bacillus anthracis infection, we would call the disease anthrax. Somebody gets a Bacillus cereus bacteria in them, we will call that food poisoning. Anyways, you'll need this table to answer at least one of the questions in the lab worksheet for today. Let's talk about the endospore stain. It's also called the Schaefer-Fulton stain because Schaefer and Fulton were the first person to invent it. The endospore stain uses malachite green as the primary stain. It gets in both the vegetative cell and the endospore. The vegetative cell, it will be the parent cell that will make the endospore. Heat is then applied as a mordant. The heat helps the malachite green get inside the gram-negative cell. Then decolorize the cells with water, and it will remove the malachite green from the vegetative cells. The cells actively growing and reproducing. After the cells are clear, you apply Safranin is the counter cell, and that will make the acid-fast negative cells stain pinkish, and the acid-fast positive cells will stain green or greenish. Here we have a picture of the acid-fast stain. Let me blow it up. And you can see two endospores right here. They are free and outside of their parent cell. And usually what happens is the parent cell simply dies, and then that releases the endospores. The purpose of the endospore, let me state that again, is simply to help keep the species alive, meaning the endospores keep the cells alive until the environment changes, and then the endospore will germinate. So those are the endospores here, and you can see endospores here and there too, but there's a difference, and that is these endospores are inside a cell. And so that cell here is making this endospore, and this endospore is almost done developing. In about 5 more days, David could get married. I know that because he's a few months younger than I am. Anyways, the cells here have part of the parent cell below the endospore, and then most of the parent cell above the endospore, with the endospore being developed inside the parent cell. And the parent cell is a vegetative cell. Any question about any of that? All right, let's move on to the flagella stain. The flagella is a structure in bacteria that allows the bacteria to swim. If the bacteria are motile, the only thing that bacteria have for giving them their motility is a flagella, or of course, several flagella. Flagella are composed of flagellin, and that's just a protein. You can state the number of flagella by giving their flagella arrangement. Let me shrink that a little. And now let me blow it up. You can have flagella that's one on only one side, and that we call monotrichous flagella. Lophotrichous flagella are, there's several flagella, but they're all located on one side of the cell. And then there's amphitrichous flagella, and those are several flagella on both sides of the cell, but only the tips of the cells. And then there's peritrichous flagella, and it is cells that have flagella everywhere. Because the flagella are too thin to be seen using standard staining techniques, we have special stains to stain the flagella and make them visible under the light microscope. Oh, it's too far. So here we have cells stained with flagella stain. And what arrangement would you call these? Because these cells are peritrichous. Let me see. This one has 2 or 3 at one end, So that would be lophotrichus. And then I think it's this one. This one is monotrichus, because it only has one flagella on one end. So what would you call these? Anyone have a guess? Is anyone there? Oh, come on. Yep, there's people there. Nobody's gonna guess. What you do when there's multiple flagella arrangements seen is you pick the highest order flagella arrangement. So these cells, which belong to one species, we would call them peritrichous. And that's because of this one, that one, and that one. Although I think there may have been one up above. Let me see if I can get this mouse to work. I'm not sure. Yeah, that's all over the cell, so that is peritrichous there too, even though there's fewer flagella than that one. So these would be called peritrichous, and the reason why there's multiple cell arrangements is that when the cell first goes through cell division and then splits into two, the cell will be like this one here, which has no flagella. And then the cell will start making a flagella. And initially, the cell will only have one flagella. And then the cell will make more flagella. And that would be probably this cell here, which has 3 flagella on one side. And then given more time, the cell can put flagella out on the other end and be called amphitrichous. And then lastly, given enough time, the cell will start putting flagella out everywhere on the cell, and those we call peritrichous. Any question about any of that? All right, know the terms of this lab? Whoop, too far. Having trouble with the controls here. Too little. Oh, there we go. So know the the terms of this lab. You can skip the references. Come on, come on. So now let's talk about the laboratory exercise. You're not going to be physically performing any of these activities, but you must understand how to prepare a bacterial smear. You must be able to understand how to stain that smear. And you must be familiar with the principles and procedures used in the various staining techniques. And then lastly, you must be able to interpret these results when using a microscope. So in procedure 1, you're going to prepare a smear for staining. Well, let me go to the worksheet now. Let me remind you, only turn in the worksheets. Do not turn in the lab module. And the reason for that is the lab module is very much bigger than the worksheet. And if everybody turned in the lab module, my space would fill up, and then there would be no more space. So students who would try to turn in an assignment could not turn it in. So the point is just what is the point? What was I talking about? The point is only turn in the lab worksheet. Do not turn in the lab module. All right, in procedure 1, you're going to prepare a smear for staining. We have 2 video clips to watch, one on preparing the bacterial smear and making it, and then one on air drying it and heat fixing it. Go through the steps for preparing a smear for staining. Any question about any of that? And then after that, answer procedure 1, number 3, this question here. What are 4 common errors when preparing bacterial smears for staining? And these are 4 common errors that students make in a microbiology lab. And remember, we're making the bacterial smear. We are not doing the stain here. In procedure 2, we're then looking at the simple stain. We have a video clip to watch on simple stains, and then go through the steps for procedure 2. Below, we have 2 smears, one for E. coli and one for Staphylococcus aureus. Read through procedure 2 and then answer the question, what is the cellular morphology, the shape of 1 E. coli and 2 Staphylococcus aureus? And let me state that E. coli is not a spiral. What's happening here is that E. coli is sticking together, and that's not a spiral. That's one cell here and one cell there. It's also not a bent rod. This is bent because there's one cell there and then one cell there. The same with this there. And then this one looks like there's probably 4 right there. There's at least 1 there, maybe 2, and then 3, and then 4 here, and then 5 there. So this is a poor preparation of E. coli. The cells are tending to stick together, and you should ignore that because all we're asking is the shape of the cells. And then this is Staphylococcus aureus, and it's very easy to determine the shape of the cell. All right, and then in B, answer the question, what is the total magnification of question 3? For procedure 3, you're performing the gram stain. We have a video clip to watch on performing the gram stain. You then can practice performing the gram stain with the virtual lab. Let me open the virtual lab and then we'll get going. So you click the continue button to begin this, what do you call that? Program on the computer to perform the gram stain.
Hi, Lab Assistant.
To,
Welcome. All yogurt has bacteria in it.
I'm just going to quickly go through this stuff. You can read it if you want. I'm going to move to the gram stain and only talk about the gram stain. I'm not going to talk about making the bacterial smear and other things. All right, on this step here, you need to read the directions, and then it's telling you what to do. You take the mouse, click the mouse, hold it down, and then bring the striker down to the Bunsen burner to light the Bunsen burner. And the same with this. It's trying to tell you with the arrows what to do. So bring it into the flame there. And this one is sterilized. Let me go back. I can't do that, can I? It was sterilized where the loop was red hot. Where it's not red hot, the loop is probably not sterilized, although it's probably clean, meaning the heating probably did kill most cells, but probably not all cells. And we're going to just skip this. And we're going to be starting the gram stain very shortly. This is making the bacterial smear that we will then use to do the gram stain. All right, now we're going to begin the staining process.
in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent. Squeeze several drops onto the sample, enough to cover it completely. Let that sit for 60 seconds. Now, rinse it under slow running water.
Okay, any questions on how to work the virtual lab? If not, I'll let you finish that and then go through the steps of the gram stain. First, you apply crystal violet. It will stain all the cells, both the gram positive and the gram negative, a bluish purple color. You then apply the Gram's iodine, and it will combine with the crystal violet to make a larger crystal structure or chemical structure. And the cells here will be purple, but they will be a little darker and they will be more of a purple color. And these are a blue purple color. And there's no real difference in this picture, but they are slightly different colors of purple. And then you apply the decolorizing step. Let me say that the best way to add the decolorizer is take your dropper and then hold your cell close to your eye at an angle so you can see the slide, and then drop the crystal violet out of the dropper on the slide so that the the decolorizer runs down. And when the blue stops running, that's when you should stop the decolorization step, and then quickly wash the slide in water, cold water. You must do that immediately because the traces of the decolorizer on the cell can continue working. And if given enough time, the decolorizer will remove the stain from obviously your hands or the slide or your clothes or anything like that. But we're interested in the slide. It will remove the crystal violet iodine complex from the gram-negative cells. The gram-positive cells should remain purple. You then apply safranin, and it will stain the gram-negative cells pinkish. Any question about the gram stain? If not, let's move on. So below we have a smear using the oil objective lens, and we performed a gram stain of E. coli and Staphylococcus aureus. Fill in the information for E. coli and Staphylococcus aureus in this table here, meaning give me the gram reaction if the cell is gram positive or gram negative, and then give me the shape of the cell. Any question about that? For procedure 3, then answer these questions. A, what is the color of Staphylococcus aureus after the primary stain is added? And it's telling you the colors can only be pink, purple, or colorless. For question B, E. coli, what's the color of E. coli after the primary stain is added? For question C, Staphylococcus aureus, what is its color after the alcohol acetone step? For question D, E. coli, what is its color after the alcohol acetone step? For question E, Staphylococcus aureus, what is its color after the counter stain is added? And then for question F, E. coli, after the counter stain is added. I'll have to go in there. Oh, never mind, I got it. All right, procedure 4 is looking at the acid-fast stain. We have a video clip on performing the acid-fast stain. In the acid-fast stain, you first apply carbol fusion, and it is the primary stain which will color all cells pinkish. You then apply the decolorizing agent, and it will pull the carbol fusion out of the vegetative cells. So they will appear clear. And you will pull the carbol fusion out of the acid-fast negative cells. The acid-fast positive cells will retain the crystal violet. And then you apply brilliant green to colorize the cells that were negative. Any question about this procedure? All right, for this procedure, answer the question here, which is, I think, up one. Yeah. Are the organisms denoted by the arrows acid-fast positive or acid-fast negative bacteria? You may, in your answer, write it above the arrow. If you don't have the program to do it, don't have the patience to do it, or just don't feel like doing it, you may write the answer either to the right of this picture or below the picture. And tell me, in the first picture, the leftmost arrow is either acid-fast positive or acid-fast negative. I'm not going to give you the answer, but only one of them is correct. Okay? And then tell me, for this cell here, say for the left picture, the topmost arrow, and then tell me if that's acid-fast positive bacteria or acid-fast negative bacteria. And then we have cells over here, and tell me if this cell is acid-fast positive, and then this cell here, tell me if it's acid-fast positive or acid-fast negative. Any questions about that? All right, in procedure 5, we're going to be looking at the 3 special stains. We have video clips for the endospore stain and for the negative stain. The picture A refers to question A under 2 here. So these questions refer to picture A, these questions refer to picture B, and then questions to C, which isn't showing here, go to question C. So question A states this is the Schaefer-Fulton stain, which uses which stain as the primary stain and ultimately dyes what cell structure that's produced by some bacteria to aid in survival. Any questions about question A? For question B, we're looking at the flagella stain. For A3, I forgot to mention, what are some examples of pathogenic endospore producing bacteria? Some is 2 or more. So if you only get me 1, it will be partly wrong. But if you get me 2, that's some. And you can find this answer in the lab module if you don't already know the answer. For question B, this is a something stain that coats long whip-like appendages which aid in motility. So what is the stain that shows for a structure that gives the cell motility? And then state the flagella arrangement type that is shown in this picture B here. Let me show B right there. And then for B3, what are some examples of clinically significant bacteria that produce flagella? And you can get that from the lab module. For question C, answer what stain this is. This is a stain that uses India ink or nigrosin and can be used to detect the presence of cell structure, which is located exterior to the cell wall. And this cell structure prevents phagocytosis. For C2 or C3, depending on how you number this, answer what are some pathogenic microorganisms that produce capsules? Once again, you can find that answer in this lab module. When we're done with those questions, we have 2 case studies to answer. A 45-year-old female is experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray and then a sputum for acid-fast bacterial culture. In addition to the culture, the acid-fast bacteria smear is made to check for acid-fast bacteria, meaning acid-fast positive bacteria. Based on the findings seen below, what is the most probable diagnosis? And you must name the species which is causing the infection. If you don't know, the lungs should all be dark, like this region of the lung here. And this region here is not dark. That's not dark. This whole region here is also not dark. And then this region up there is not dark. So that's what's wrong with the lungs. And there's the sputum smear. And there's the cells stained with the Zeal-Nielsen stain. You can look up in the lab module what the Zeal-Nielsen stain looks for. And that's the results that you're seing here. Moving on to case study 2, a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis, and that is seen here. And that's actually a very serious disease. A routine culture is done. The gram stain is ordered. The bacteria only grew anaerobically. So that's one hint. And based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. Here is a picture of the gram stain. And here is a picture of the Schaefer-Fulton stain. If you don't remember what this stain stains for, and you can't figure it out from the picture, the Schaefer-Fulton stain is mentioned in the lab module. All right, when you're all done, write your answers in the worksheet and then submit the worksheet by Saturday, 11:59 p.m. Are there any questions? If there's no questions, I'm going to stop sharing my screen, stop the recording.